Hi, I'm Meredith Blackwell. I'm at the Mycological Society of America meeting at Minneapolis. The year is 2019. And we're here to do interviews for the Oral History for Mycology project. And right now we have Linda Kohn and Jim Anderson. They are two mycologists who met in mycology. And I'll let them tell you how they met.
Yeah.
So,
you
go.
Kick it off,
Okay,
Linda.
so
Tell the first day story.
Yes, I met Jim on his first day of graduate school, and he was nervous, and he was studying Alexopolis and Mims. and I said, "What are you doing here?" Because he was occupying the room that I had with great difficulty secured for preparing for a course summer course in mycology at the University of Vermont, and there he was. And the rest is history.
Yeah.
But we did run into each other. I told him that he needed to learn more about fungi and that he should go to the University of Michigan Biological Station, and he did. And that was the next place we met.
And you didn't know that I would, but I did. And I contacted Bob Schaefer, who said, please join us. You know, there's no official status of any kind, but I didn't even have to pay any fees, and I learned so much that summer because I was just a participant in all the field work and the classes. And it was marvelous. And then Linda came to the biological station for the last 2 weeks of the summer.
As an independent investigator.
Yeah.
And I have to say that Bob Schaefer's course was so formative for me. Carrie O'Donnell.
You did it earlier, of course, in 74.
And Tina Gilliam was my TA.
Oh.
We became friends, and Carrie O'Donnell was in that class. We had a superb class, and we competed and goaded each other, and that made it all the better. We learned so much. But Bob would put out all the mushrooms and we were collecting in Alexander Smith's old localities, rich with fungi, and Bob would label them, and I would sit after hours keying them backwards in Bob's really wonderful key. He had written a key for us, and I learned so much.
And I think I got that key when he gave a presidential address.
Was
to
it the little
society.
key of
the blue book? Yeah.
microscopic
well,
characters?
this was photocopied. But he handed it out with his presidential address and a classification, I think.
Because the emphasis was on microscopic characters.
Right.
And it was funny, but the labels came up the other day because people were in the symposium yesterday morning, because people were saying they had copied those labels to use at the Mendocino foray.
I was delighted to see that.
So that was interesting. Well, I think I first met you two when Linda wanted her picture taken for the MSA newsletter.
Okay.
And did both of you win the graduate fellowship the same year?
Linda won the 1 year, and I think I got it the next year.
Okay, but for some reason we took your picture together, so that's why I was thinking you'd got it the same year. But because they do give two, at least now they do.
Yeah.
But anyway, I remember this was in the days when you had to take a photograph, and we all had probably Kodachrome film.
Yeah.
So you had to get the slide developed and then get the print made. So it was a lot of trouble, Linda. But we did it. Anyway, so both of you have gone on to have great careers in Canada.
Yeah.
You were at Clemson for just a short period
I was
of time.
there for 2 years, and I'm delighted that Julia Kerrigan is my successor.
She's really worked with her in her group. The amateur group that she helps with is really
I
learning
met them.
a lot of.
Actually, I was there. I was invited to give a talk for a new program to recruit and retain female.
And guess who was there and heard you? Me.
Yeah.
I drove
well, you
from
traveled
Columbia.
together, didn't you?
We had a great time together, Meredith.
Yep.
It was terrific.
So it was very nice. And so, but anyway, why don't you each tell me one at a time, whoever wants to go first, where you were born, and what kind of family were you in? Were you in one that was very supportive of mycology? I know we were talking to Mel Fuller the other day, and his family was not.
Yeah.
But
anyway.
Yeah, I'll go first. I was born in Madison, Wisconsin, moved to Rhode Island, so I mean, at the heart of it, I'm a New Englander. My family knew nothing about science. There was no support whatsoever. It was totally on my own, although I have a history of it because, as it turns out, my grandfather was a professor of engineering at the University of Wisconsin, but there was not much academic tradition. I think my parents thought I should be doing something else
Physical therapy,
yeah,
medical school.
something more reliable. You know, becoming an academic is highly uncertain. Even then, you know, it was tough to get a job.
Yeah.
I mean,
Jobs weren't even advertised
No.
much then.
yeah. And when I interviewed in Toronto, they said, there's no way you're going to get this job because you're an American and we've got a lot of competition. It's a lot of trouble for us to hire a foreigner. But by the end of the interview, they said, you know, you're
might
going to get
get
the job if you finish on time.
Yeah. So what
So
about?
anyway, no support at all.
Yeah.
It was all kind of just a new thing in the family.
Had your folks been to college or?
Yeah, they had been to university. They were both in the Navy in World War II, my mother and my father.
Wow.
And
That was very
so,
hard.
yeah, no, that was great. And they were happy about it all, but I think they were maybe surprised and didn't ever really understand it.
But they did live to see you get a job.
Oh, yeah, they did.
Good.
Yeah.
And to marry me.
Yeah. Which we did after we both started.
So I was born in Schenectady, New York, which is a lodestone. It's a lodestone for spelling.
No, I
Don't even try.
No, it's good.
Not
I
me.
can
I'm a
spell
bad
it.
speller.
And so my father was an engineer at General Electric, starting in World War II, and designed armament systems. And so he had gone to MIT, and so he came from Chicago, and like he was in the top tier of his graduating class, and they all went to MIT, I think. Anyway, so he was a very smart man. He was he you know, I would bring him books like by Lauren Isley and things about science, and
Oh, I
he would
remember
read
reading
those
those.
things. And he and Jim forged a wonderful friendship.
Oh, yeah.
So
Yeah.
he compensated for that thing that was maybe missing from Jim's
Jim's
father.
Yeah, and my parents were gone at that time, so Julius was like a second father.
Our parents got along very well, and
Yeah,
my
they did.
mother went to Marjorie Webster Junior College, and she worked as a draftsperson at GE, and that's where she met my father.
Oh.
Now, I just want to say she wanted to go to Skidmore, where Jim's mother went, and they said, well, she interviewed, and they said, we're sorry, but our Jewish quota is filled.
Oh my gosh.
She was qualified, but
Yes.
not accepted.
So that
So there were
I
quotas.
came from that situation in many stages of my life. So I've lived in Schenectady, in New Jersey, and that was also had the place we lived was red light. Kids would tell us they couldn't play with us because we were Jewish. So that
my
was
gosh.
really weird. Anyway, and we wound up in Burlington, Vermont, and that was wonderful. Everybody was happy.
And you were accepted.
Yeah.
Did
Yeah.
people
Oh, yeah.
worry about
And
religion at all?
Well, I went to summer camp there before we moved there, and I just had great friends and of all sorts. And but I love Burlington, Vermont to
We
this
still
day.
have strong ties
Yeah,
with Burlington.
yeah.
We're really binational now.
We are. We
So
have 2 passports.
Good.
We vote in both
we
countries.
feel very Canadian.
This
Well,
had a big impact on my life
I'm
because
sure it
of
did.
also events from my father's was German, and a lot of the family was. So this is very imprinted in my identity.
Yeah.
These things. So anyway, yeah, you love my father.
Oh God, it was
And
great.
he
well,
really-
he's a scientist too,
Yeah,
so we
yeah,
had a lot
so-
to talk about. Even, you know, we used to talk about physics and things like
They
that.
had an argument in a sports store about where the Celsius and Fahrenheit scales converged, and people turned around and they were like, "Who are these people?"
Okay.
But
so
I collected mushrooms and inflicted them on my family, and they
did they eat them or
yeah.
just?
Well, I came back with a mother load of chanterelles once. And they were skeptical. I stored a lot of things in the fridge, including poisonous ones that I wanted to keep. But I cooked up those chanterelles. I converted them.
Yeah.
They learned to trust me.
Yeah.
Yeah. So you were at Clemson, you were at your Toronto.
Yeah.
So how did you get to Toronto?
Well, you better talk about that, Linda.
Right. So Jim was hired in the
Yeah,
in
yeah.
the normal.
1980.
He was
Even though
hired.
he was an American.
I was 27 years old.
Yes.
Wow.
Yes.
No postdocs in
No
those
postdoc.
days.
Well, that's a
There were, but I didn't do one,
Okay,
which was a
you
big
were exceptional.
lack in my life,
Did
really.
you?
Yeah. No,
I
I
think
didn't
you
do a
overcame
postdoc.
it.
Well, you were offered one,
Yeah.
but you got this job.
Yeah.
And so I had a job at Clemson, and we decided to get married. And Dave Malik in Toronto applied for a strategic grant. It's a special kind of
I
grant
remember
to
that.
support
Yeah.
me. And he got it. And so I went and worked on mycorrhizal fungi with him. But then that grant was exhausted, and I patched together. So I've worked with the Faculty Association at the University of Toronto and I brought to it an experience with every kind of precarious employment that women confront in universities. I experienced that en route to getting a tenure stream faculty job.
And you were married already.
We were married
Yes.
already.
You'd been
But
married.
the great thing was Linda got her position independently of anything I could do.
Yeah.
I actually I applied for a NSERC, our granting agency, had a program, the University Research Fellowship Program, and it was meant to bridge PhDs through a time when there weren't many jobs, but people were going to be retiring, and it was meant to
keep
keep
you there.
us
Canada.
keep us engaged in Canada. And it wasn't easy, but and you had to be put forward by the department, and I was able to apply. Now, this was not just for women, it was open to engineers, mathematicians, biologists, all kinds of people, and I won it. And so I created my own job. It gave me 5 years of my salary paid by NSERC. The university paid my benefits and an instant research grant. And so I created my own job. I got tenure. The NSERC stopped, it tapered off, and the university paid my salary, and I was on the tenure stream.
Yeah.
But I created my own job.
Yeah, it was great.
Okay.
Saved our.
Our marriage.
We wouldn't be married if we hadn't gotten that. Probably something would have happened. There would have been a divergence.
So
Who knows?
insert and the Canadian.
We're very.
Fellowship.
We are
grateful.
happy taxpayers, frankly.
You know, we had like 38 years of continuous funding in Canada. So,
Wow.
you know, maybe it wasn't as high as it was in the States, but we had continuity.
Well,
We
you
could
know, though, you
plan.
didn't pay lots of overhead.
We don't pay overhead.
Yeah, and so they're not really that different.
No, it's not that
Yeah.
different. Yeah.
And the other thing is, whereas we do 15 or 20 pages, you guys fill
5.
out a form.
5. Well, it's a little
Okay,
worse
it's
than
more-
that now.
okay,
but
it's better.
But it's not much compared
Okay.
to an NSF
Yeah.
proposal.
Okay, so let's talk about your research, because you've both done outstanding research and been invited all sorts of places to give your results. And some of it you've done together, but,
Yeah.
but independent. You've
Yeah.
been independent too.
Yeah, yeah.
So why don't you talk about that?
Yeah.
So my specialty was discomycetes, the Sclerotiniaceae, voracious plant pathogens. Sclerotinia sclerotium is omnivorous on any broadleaf plant.
Yeah.
And starting at Cornell was really good for me. So a big thing in much of my career was collaboration with plant pathologists. And
I should say, we neglected to say where you got your PhDs. And
Okay.
you got yours with
Korf,
Korf.
yes,
Korf.
Richard
And-
Korf at Cornell in the Department of Plant Pathology.
And so
And-
you picked up plant pathology.
Oh, yeah.
And
And me
Jim?
with at the University of Vermont with
Right,
Bob Ulrich.
Right.
So, so I really enjoyed working with farmers and plant pathologists, and I have to say, so there was a huge controversy with Sclerotinia sclerotiorum. There was one person, an eminent male person, who who thought there was one species. There was Sclerotinia minor, Sclerotinia sclerotiorum, Sclerotinia trifolium, and he was adamant that they were all one species.
That makes it easier.
Yeah.
Well, for him, yes. And so there I was, small, young female person challenging this because the kind of data I was collecting indicated that there were three. And this culminated in molecular data too. And so, and I was lucky because the plant pathologists, that community adopted me as I was like their mascot. They were very kind. Yeah, people like Georgia Bowie and Jim Stedman.
Oh, yeah.
Just really lovely. Marty Dickman, who passed away. He was a great friend of mine. They were so welcoming, you know. It was, and so it helped me enormously, and they put me forward at meetings and things, and it was good. It was very good. So I maintained this work with, people like Mark Cubetta, this work with plant pathologists. Pretty much until fairly recently. And I could find graduate students who are interested in that kind of work. So that was great.
Yeah.
So Jim and I, our work was pretty separate. He was working with
on armillaria.
Yeah. I remember one of your papers very, very early about polyploidy or
we started out looking at the mating system because it was unknown at the time, and a Finnish worker had done a key piece of work that laid the groundwork. But I sort of did the genetics and came at it from that point of view. And in those days, the markers were not DNA. They were. I made auxotrophic mutations at great labor and used them to trace where the nuclei were going. And we showed that the fungus is not dikaryotic, but it's diploid in its vegetative state.
Yeah, that's what it was.
And we also learned about intersterility barriers and biological species. And I had no idea at the time how good a project that was. I was just immersed in it as a graduate student, and so I took that forward into a faculty position.
I sent him material too. I sent
you did among many collectors.
From South Carolina.
From Vermont.
I
put out this
earlier,
call to mycologists to send spore prints.
We used to do that in the newsletter. Is that what you did?
No, I sent letters to a whole bunch of people, and everybody responded,
better.
and I got a great
Where did
sample.
I send you material from?
It was from Ringwood. And,
That's right.
And
It was from Cornell.
It was
It was Cornell.
Armillaria melia sensu stricto.
Yeah,
it's the
it was really
type
good. He was
Armillaria melia.
Yeah.
But I learned about all these other intersterility groups, and people have since done the taxonomy. I wasn't a taxonomist, so I didn't do that. And it went molecular, and it was just great. But then there was a 2.5 decade hiatus. Linda and I worked together, and I'm really an experimental kind of guy, not systematics and evolution, and we kind of brought that all together with yeasts, and Linda and I worked together.
Except there was a great eureka moment in my work on Sclerotinia, and it's a paper in phytopathology. And we were using RFLPs. We made beautiful southern blots. It seems quaint now. And so I found it's a non-LTR retrotransposon, and it's and we used it as a fingerprinting element.
We worked together on
this
project.
And we discovered there were clones in Sclerotinia sclerotiorum, and they had unique fingerprints. It was one of the great eureka moments of my career, my research career, and Jim and I shared that together.
Yeah, but then quickly, we needed a system that was more contained, and I wanted to be able to do evolution experiments over the course of a few days or a few weeks.
Talk about those. Those are really neat.
So we got into experimental evolution, and we started with evolution of drug resistance because it's tractable, and we knew the candidate genes, and we could take Candida albicans, sensitive to an antifungal, and over the course of time, through serial passage in the presence of the drug, we found resistance types.
So I have to say two things. So I think this fit doing experimental evolution is evolution in a test tube. You have a fossil record in the freezer it's incredible. And I think this fit a need that Jim had.
It
was
because
really fit him perfectly. But we also had the perfect student. We had Leah Cowan, who I hired. She was an undergraduate and wanted a summer job. She was an undergraduate at UBC, and I just want this in here because she wrote to the department, and it was unclear what she wanted. She was an undergraduate at the University of British Columbia, and people thought she wanted a graduate school. She wanted to come to graduate school, but no, she wanted a job. And I said, send me your grades. She did. They were great. And I hired her to work in the lab with Ignacio Carbone, and he was doing a lot of sequencing. And she came, and oh my God, she was wonderful. She decided to work with us. She applied for an NSERC graduate fellowship, and she got it. Oh man, that was a great.
She got 2 supervisors.
So there's
for the price of one.
There's some things for people to know. Like if an undergraduate writes to you, get more information from them, because Jim Cronstedt at UBC later after her thesis exam, he said, "Why did I miss you? How did we let you go?"
I had one like that, New Wynn, who's now at Hawaii.
Yeah.
And New came to the lab when he was a freshman. And he had written me an email, and I later asked him, why did you choose my lab? He said, you're the only one who answered my email.
Wow, wow. Yeah,
so then he went on to work with Bruns, and he's now in Hawaii. Or after Kennedy, too. He did a postdoc. But I was going to say, Kohn has been mentioned several times here at the meeting because she continues to get
She's a leader.
She's a superstar.
She's a leader.
And Joe Heitman has talked about this project he's on with her.
He's actually been a terrific mentor.
And friend. But she's
so here's another story about Leah. So Leah and I were at a Candida meeting and sharing a hotel room. And we had the important talk. And she said, what do you think I should do? And I said, well, what are you thinking? And she said, well, maybe science journalism. And she said, what do you think? And I said, I think you've been in a small lab. I think you should go to a big, fabulous place with the best person possible. And she did. She went to work with Susan Lindquist at MIT. And she rose to the occasion. And that helped her enormously getting a great job in Canada because, that was a great credential. So
that's a big lab with dozens of postdocs.
Very impersonal.
So you have to make your own lab.
You have to make your own way.
Okay, so back to your microcosm studies.
Okay, so we did this whole thing with Candida, and it was great because we discovered the molecular mechanisms of resistance, which were known from other systems, but we identified them in ours. And then we went back to patient populations.
Well, we had to collaborate with a couple of infectious disease physicians to have access to them. And we had 8 trajectories to drug resistance. For example, becoming, these were replicate, it was a beautiful experiment, 12 replicate populations founded from one single cell but reared separately. And so so there were, for example, one line would become very drug resistant and maybe stay there. Another would do that and then drop, and some were mediocre. So we had these 8 trajectories, and then we had a panel of clinical isolates from people who'd been treated with fluconazole
and
had some resistance. And we found the same trajectories in clinical isolates.
In some cases, the fungus would
another eureka moment.
would increase the efflux of the drug, and in other cases, it was the target itself that was modified, and other things going on with ergosterol metabolism. So there are different ways of getting to the land of being, resistant to an antifungal drug.
And we took that further, collaborating with systems people at MIT.
But then that wasn't enough, and we needed more. We needed more of a model system. And remember, I had a grant from the Medical Institute in Canada, CIHR, and they advised me, you need to work more on Candida. We had a Saccharomyces cerevisiae component, and I went with cerevisiae. I ignored their advice because we needed that. We needed the genetics and the genomics and the support. So that was the next phase. And we went into more general questions of adaptation, divergent adaptation, and how that may impact on reproductive isolation. So that was a real light bulb moment, wasn't it, Linda? We said, nobody's done this, oh my God. So we took from a single cell, we made replicate lineages and adapted them in different environments. And then we made hybrids and showed the hybrids were all reduced in fitness. So there's something going on, isolating those, and it really mimics what we would expect to be the first stages of speciation. We didn't make new species, but we're following the process of incipient speciation.
And you should mention Jeremy's.
Well, Jeremy Detman he came from the Taylor lab.
Yeah.
And he did a postdoc with us, and my God.
We were in heaven because he's a wonderful experimentalist, and he's wonderful working with students, and it was just a pleasure.
And writing, it was fun.
Anyway, but he measured negative epistasis.
Yeah.
You know, you've heard John Taylor's group talking about the Dobzhansky-Miller model and conflict when you bring different genomes together through hybridization, and Jeremy actually measured negative epistasis.
We had the means to measure fitness because
I was
back in
gonna
those
ask how
days
you
we
did it.
we barcoded the strains. You know, we did transformation and put in artificial barcodes, which Jeremy, we designed them by flipping coins. So 2 coin tosses per nucleotide position, no retakes, no cheating, and we'd design a 20 base barcode that we would put into the fungus. And so they can be quantified. And we did that in a very low-tech way so we could do hundreds of samples simultaneously. And basically, in a lineage, measure the number of doublings. So we could compete multiple strains in the same arena and see who's going up,
evolutionary
who's going
drag
down.
Races.
Yeah, and it was actually more accurate than methods that are currently being used. Perhaps more labor intensive, but so it gave a really good measure of fitness. And not only that, we could quantify the amount of fitness that was just due to having accumulated different determinants of adaptation, and also the component that was due to the negative interaction. We could separate those 2 components for the first time because unlike flies, we could compete the progenitor with the evolved, and we could make hybrids between the lineages and back with the progenitor, and there were some really fun things you could do in all those comparisons.
Is anyone taking this up and using that system?
Oh, I think they've taken it up and it's gone
So
ballistic.
that paper is in Nature, and it was one of the first times that epistasis it was quantified negatively. It's cited in that way, you know, as an example.
Yeah, because we started with one cell again, and all these different effects were, you know, the effects of adaptation directly. And we also identified the mutations, so we did all the whole genome sequencing later. And
the network of interactions is very complex.
Yeah.
But it's
but we identify some key players. So when you get this allele here and this allele here, and you put them together, they hate each other, and fitness goes down because a lot of these have to do with major regulators or major either of gene expression or cell physiology. And so when you start making these major changes and you put them together, there's a lot of potential for bad things to happen. You know, like when you load new software on your computer, EndNote doesn't like your Word program, and they don't do well together. Who hasn't experienced that?
Okay, so you both had...
We need to talk about collaborators too, because
Okay.
I think that's really...
do mention
so
your
Jim
collaborators.
has you're the 3 amigos there.
Well, yeah, that goes back to Myron Smith, who was my first PhD student, and we still have a great friendship and collaboration, and we've been back to revisit
The
the
humongous
fungus. Remember that?
Oh, definitely. I've even seen the U-Haul.
Yeah.
I know, it's
Yeah,
cool.
well, 3 decades on, we went back with the question, is the thing still there? Yeah, of course it is, but we had to answer that question. And also we did the whole genome thing and looked at the mutation dynamics. And what we found is that the fungus is extremely stable genomically and genetically. You can think of a spectrum of mutability where you've got cancer over here, which is spinning out variants at high frequency. Armillaria is the example of rock-stable propagation.
So
We
the
found
entire-
mutations, but it's very, very stable
In the entire phallus.
entire individual.
Yeah.
And the mutations that do happen, we can trace them to specific spatial sectors. So it looks like a petri dish where you see
You've
this
got
sector and
sectors.
that. We see the same thing over a kilometer-sized fungal individual in the forest.
It's, and it is sort of like a phylogeny,
It's a physical
because,
phylogeny.
Yes, it is. You get it? Yeah, yeah.
Because a branching mycelium is itself a tree of life.
Yeah.
Physical position correlates with relatedness. Time back to a common ancestor.
And Jim actually went to Dick Korf's Island, and there's plenty. Actually, if you look anywhere, if you plant a potato or pound a poplar steak in the steak and potato method, you're going to pull out armillaria.
Yeah.
You will get
Yeah.
rhizome. So Jim's actually, he had a student who looked at the distribution of mutations in
Yeah.
the network on X Island.
In another individual,
Yeah, it was
which
another.
we knew about that individual 30 years ago, and it's still there. So yeah,
It's at Dick Korf's Island.
yeah, it occupies about 2/3 of
Sacred
Dick
territory.
Korf's Island, and has the same territory now as it did 30 years ago.
So Jim has a really nice paper out
Yeah.
in Royal
Proceedings
Society
B,
B,
yeah.
Royal Society B, in which it's well presented there.
Yeah,
It's
yeah.
very,
yeah.
very.
And it was the result of maybe 5 weekends of work over 4 years. The 3 amigos got together. No students.
Who was the other one?
Myron
Johann
Smith
Brun.
and Johann Brun.
Oh, okay.
At
Oh, yeah, of
Univer-
course, I knew that.
Yeah, Missouri at Columbia. And we did the work, no students. We did the lab work. We did the data analysis. And so that was really fun to get right back in on the front line of something. But it was a hobby that worked out really well.
Yeah, it really created a
Yeah,
stir.
no, it was
hit
neat.
all the popular press. And
Yeah.
It was really neat.
Oh my God! It was astonishing. There was
the
a
recent
day.
one,
There was actually the big. Then in 1990, it was April Fool's Day in 1992
When the big thing hit the
paper
fan
came
and,
out. And
yeah.
you remember we had cubby holes for our messages.
Yeah,
Remember
yeah,
that?
yeah, yeah.
Jim's was overflowing.
said, Jim,
I looked at them. They were
you
from
better
the
get
New
back
York
there.
Times, the San Francisco.
Well, and that they were all
My
out
mother
over-
was cooking dinner, and she was listening to All Things Considered, and suddenly Jim's voice came on,
And
and
she
she
recognized
apparently
it.
threw her skillet up in the air and screamed, "Jimmy!"
I even got a call several years later from Outback Magazine.
Oh, yes.
They
yeah.
were very interested.
But the recent one, Meredith, was also picked up.
Right. No,
Science
no,
did an
I
article
heard
on
it.
it, New
I
York
heard it.
Times.
Jim was
yeah, it
standing on the dock at the cottage getting interviewed on his cell phone.
Yeah,
It
trying to
wasn't as
fend
scary
off the
as
dog
it
and-
wasn't as the dog was
Yeah.
barking. Anyway,
So what other collaborators did you have? You can't top that
so
one.
Tron, I have to
Yes,
say, the
Yeah, Tron.
Norwegians, he's my brother from another mother.
Yeah.
We have this mind meld, and it's extraordinary. And we've been all over Norway, and I have to. And he came and did a sabbatical in Toronto. His student Arne Holst Jensen, who's now a faculty member,
He worked
worked in our lab.
for a year.
And they're extraordinary, it's this Linnaean knowledge of nature, sort of. They know plant phenology so well, and they have such an interest in finding new, working out a whole taxa, you know, and getting all the but they can do it because of their skill in the field. And it was such a wonderful, it was a pleasure. Trond and I drove all over Norway. We were once in cloudberries. We were looking for a species. It was a species, a new species. Trond and Arne were describing on cloudberries, and there was a bicycle race going on the highway next to us, and they were yelling at us because they thought we were prematurely collecting cloudberries before they were ripe, and we were taking their cloudberries, and we were berated for. But we were all over the place, and
Linda and Trond used to bicker like an old married couple. You know, they knew each other that well.
We were once in a restaurant in Oslo on the Norwegian National Day having kind of a stimulating argument. And somebody jumped up and yelled at Trond for speaking English on the Norwegian National Day, and Trond told him where to go. But it was wonderful, you know.
And we were just back there in June.
Yes, we were celebrating Trond's retirement and
really?
70th birthday. It was fabulous, and we spoke at the botanical garden in the department. We were at a fabulous party in the mountains, and we got to revisit all this great.
Yeah, no, that was great.
Oh my God! So that's one of the great things of my life, is that wonderful collaboration.
And you talk about Trond's retirement, but both of you have retired.
Yeah, we have. Yeah.
Why did you decide to retire now? Because you had so much going.
Yeah, you know, I think we wanted to retire with some gas in the tank still.
Okay.
And also you get to the point where, you know, I've been doing this since I was 27. And maybe it's time to switch it up and turn this position over to a younger crew.
Yeah.
And also now we can be freelancers. We can do
So we have
science.
this great thing going with yeast in nature.
Oh God,
and
yeah.
it's and we have field sites on our campus, and we still have a lab for a couple of years, and so we can do that, and we're finishing up some.
Yeah.
We've finished up some papers.
I mean, we weren't that young, you know.
No, I'm old enough.
I was the normal retirement age, which was 65, and I'm now pushing 67. And Linda, you were 67 when you retired.
68.
68, was
I'm
it?
69 now. It's
and,
okay.
Yeah, it's okay. A lot of people are hanging on, but we think that maybe there is a point where people should retire.
Yeah, and I like to travel, so I feel much freer and able to do that kind of thing.
You do what you want to do, and
yeah,
you're still.
when you're teaching, one missed class or two missed classes.
It's a disaster.
Good.
Yeah.
Not good.
Yeah. So
we're happy not to be preparing lectures and
I
the course
got
syllabus.
very tired of that because I always threw away all the old.
Yeah, you were
every
always.
year.
Yeah, you put everything into it, and it's tiring.
Also, running the franchise that is a research lab, you know, it's a big job, and we became seeking grants, seeking great personnel.
And realizing the minute you didn't get a grant, everybody was out, you know.
Oh yeah, yeah.
And especially in the U.S., if you have foreign postdocs on certain kinds of visas,
They have to go.
Yeah.
Yeah.
Go home.
But anyway, so we wanted to kind of end on a good note.
Yeah.
And
Well, you certainly did.
yeah,
Yeah.
it felt
But
that way.
the last when we finally started working together, the yeast stuff has been so much fun.
Yeah.
We
last
had
years.
undergrads working on this work, the ecological work with yeast.
And it was tractable and doable, and it was kind of like a barn raising, you know, with undergrads. They were great.
Yeah.
They would argue about the best way to do things, and no, I don't think you're testing the hypothesis you thought you were testing. And
we had an extraordinary visiting master's student from Munster, from Germany, and he needed a component of lab for his degree in another lab, and he came, and he was great because he elevated
whole discussion.
So I think that I also recommend that is a great thing to have someone come in. It can shake up the students, the local students, you know, and it
And
sort
we
of
also,
upped the
we
bar.
send our
yeah.
students on a mini sabbatical.
Yeah.
So graduate students were,
That was one
we
of our
required them
innovations.
to get out there for a few months and go visit another place and
Yeah.
get on that steep learning curve.
Yes.
And that was, I think that has helped people a
Yeah,
lot.
the mini sabbatical.
And then
was
they come back, you know, they've obviously experienced something new, and it
Right.
has a real impact on the work
Yeah.
they're doing.
And then they appreciate you more.
Yeah.
So
Yeah.
the other thing, yeah, like Brett Couch was with Michael at Cornell.
Yeah.
And that helped, that was great, and took a great pop gen course at Cornell.
With Chippequadra,
And it was beautiful. So we're firm believers in sending graduate students out for sojourns in other places. Yeah. Yep.
Well, very good. I think unless there's something.
But so Meredith, you've
Covered
been
a
a
lot.
great inspiration
Yeah.
for us too.
No.
Yeah.
We've always admired
You
you.
have. I remember I remember my first MSA meeting, and I said, well, you know, thank you, Dr. Blackwell, for taking my application for the MSA. And you said, forget the Dr. Blackwell. It's Meredith.